2-3 papers. But then, as time passes, he starts realizing now that the combinatorial problem becomes large, and you have to use computational techniques to solve it.
Okay. Hello, everybody. Thank you for showing up. It's a pleasure to have to introduce today Professor John Goutsias from Biomedical Engineering Institute, and also from EE and Applied Math and Statistics at Johns Hopkins University. John got his degrees from USC, after his degree, of course, from National Technical University of Athens, and joined Johns Hopkins in 1986. He's been there since, and where he's currently, as I said, a professor, and he's done a lot of very interesting stuff in a lot of interesting research in imaging, nonlinear imaging particularly, and I guess over the last 5 or 6 years he's been doing some excellent work also in bioinformatics and genomics in general. He actually was the second organizer of the genomics workshop that started here, if you guys recall, in Raleigh. It's a pleasure to have him here, and he'll talk about some of his biochemical reaction systems. And thank you for coming.
Thank you, Hamid, for inviting me. And thank you for coming also to the talk. The subject of the talk is techniques to do sensitivity analysis for biochemical reaction systems or networks. And basically, it's kind of a review talk, which I will try to pinpoint to you what the problem is, how it has been addressed, and what are the challenges that remain in this area, and of course why we want to do things like this. And I understand that most of you are not familiar with biology and reaction systems and things like this.
Guilty as charged.
Yeah, you're guilty. I know that. I don't know about the rest of the people though. And I will try to give you some background so you can understand what I'm talking to you about. Sensitivity analysis, it's a very general subject that applies to many areas of research, and as a matter of fact, there is a very big big part of work that has been published in mechanical engineering journals, also in economics journals, and now in biology. And basically what sensitivity analysis tries to do is that there is a system that is usually described by differential equations, which basically depend on some parameters, and you try to selectively perturb the values of these parameters and see how the output of the system behaves in these changes. And depending on what you find, you can detect which parameters of the system are most crucial in shaping the response of the system, whatever the response is. So in economics, for example, there will be very complex differential equations that will describe, let's say, the economy of a nation, which will depend on various parameters, like, I don't know, the stock market, the resources, the water resources, the whatever. And then you will perturb these parameters and see what perturbations affect the behavior of the system the most. In biochemistry and in computational biology, as you're going to see, this is extremely important because it has the potential of giving us a wealth of information about what components of the biological system that we're interested in are most responsible in shaping the response, which the response usually translates in very serious stuff, like, for example, fate of cells, cancer, various other diseases, robustness of the system to drugs, how drugs can affect the behavior of cells and things like that. So it's very important. So we say that parameters of the system that produce the highest output fluctuations are associated with the most influential reactions in a biochemical reaction system. And there are basically two types of analysis that people are using. The first one is based on very simple derivative-based deterministic tools, and I'm going to explain to you in some of these tools, which are very simple to understand. The second set of of tools are based on stochastic approaches and lead to stochastic sensitivity analysis techniques. In order to do sensitivity analysis, you have to basically specify three things. First of all, you have to have an application, of course. You have to build up a mathematical model for this application or for that system that you're interested in. You have to pinpoint which are the system parameters of interest. And of course, you have to determine what are the system outputs that you're interested in. Okay? So the model is going to be used to do the computational analysis. The parameters that you identify are the ones that you're going to perturb. And the system outputs are the ones that you're going to measure. Okay? And see how these measurements are affected by variations in the system parameters. There are many applications. And here are some. Sensitivity analysis can give you a lot of information about the robustness of the system, because if you realize that there are parameters that no matter how much you change them, the output does not change much, then that means that the system is robust. As a matter of fact, biological systems, all the biological circuits inside our cells are extremely robust to many different environmental conditions and variations, because if they were not, we're not going to be alive now. We're not going to exist as species. Okay? Fragility is extremely important, because then if you identify one or two or three of some parameters that very small changes produce large variations in the output of interest, then that means that the system is very fragile to these parameters. And biological systems also are very fragile to certain parameters and certain conditions. And they have built other circuits to control these parameters and these conditions, so we don't get catastrophic results. But these are extremely important components of the system, because this can give you a lot of information about how to intervene by means of drugs, by means of therapeutic interventions, by means of all other things, in order to correct the system in case that the system is leading to a disease or something. Also, many times from data that you obtain from experimentation, you try to estimate the parameters of the system by using estimation techniques. So it doesn't matter to estimate much. You may not care to estimate parameters that the system is robust to, because no matter what the value of these parameters is, the output will be the same, more or less. But you are very much interested in estimating the parameters that the system is fragile to, and you have to spend a lot of time estimating these parameters very precisely, because small variations will create large variations in the output. So this is very important. And this also leads to the notion of model simplification. Biological systems are very complex. We don't know much of these systems, and it's very difficult for us to come up with a very detailed model for these systems. And when we come up with detailed models, the models are so complex that we have difficulty analyzing them. So by using sensitivity analysis results, we may simplify the models by creating a simpler model that will be function of the most important parameters, and we can focus our research on an analysis on those parameters. Identifying critical components, I told you before. Prioritizing research. If you find that part of the system is important, then as an experimental biologist, you are going to spend time and effort and money in studying that part of the system. You don't care about the rest. Selective perturbation is very important. Many experimental techniques collect data by perturbing biological systems, either artificially or naturally. Natural meaning that you get a human being that is healthy and a human being that has a type of cancer. You extract information from cells, you image this information, you get you go through experiments and you get numerical data, and you compare this data to see what's happening. So this is like a form of perturbation and intervention to the system, as I explained to you. So these are some of the... So as far as we're concerned, that's very easy to understand. If you're an electrical engineer, you don't need to come up with details. We're talking about molecular, a number of species, molecular species, which will be N based on this talk. These molecular species can be proteins and different proteins, or can be other components of the system like messenger RNAs and DNAs and all kind of things. But let's say that there are n different molecular species in general, and these species interact with each other, and they produce by means of reactions, which we're going to assume that there are m reactions. And based on this talk, just to be rigorous, we're going to assume that this is monomolecular or bimolecular reactions, which are basically most of the reactions that occur in nature. Monomolecular means one molecule is transformed to some other molecule or molecules by itself. Bimolecular means that two molecules collide with each other and their collision produces products. So you have the reactants, which will be one or two, and the products of the reaction. So you have n molecular species, which will be the reactants, that they interact with each other by means of m reactions. And in biochemistry, based on some assumptions and simplifications, you can describe the system by a set of differential equations like this. So this is a vector, and this vector denotes the concentration of the molecular species in the system in time t. So this is an n-dimensional vector. This is a first order equation, so it's a very simple equation. Okay? However, there are some complications here. This rho is also a vector. It has dimension m, so it contains components that they depend on the reaction, not on the species. And it's given by means of a function on the concentrations, which is if the reaction m is monomolecular, this is just the concentration of the reactant molecule. If the reaction is bimolecular, is the product of the concentrations of the reactant molecules multiplied by a constant kappa m, which is characteristic to the reaction, and this is called the rate constant of the reaction. These are very classical equations developed several decades ago. Okay? So this is basically a system of differential equations. This is how you should see it. And as far as this talk is concerned, the parameters of interest will be this rate constants. Okay? The mathematical product of the system is the vector q, which is the concentrations. However, so this is the model. These are the parameters. And now we have to come up with the output of the system, or the outputs of the system. Of course, we can consider as the output of the system as being the vector q. But then, you know, usually there are many species in a biochemical reaction network, and this can be overwhelming, keeping track of how all these q's will behave, and maybe also irrelevant. Okay? So let me show you an application which will help you understand what the output will be. So this is a model for something that we all have in our bodies. Actually, all species on Earth have it on their bodies. This is called the MAPK signaling cascade. Is a mitogen activated protein kinase. This is the biological name of this. This particular circuit shows up in various signaling parts of our cells, but it came from a signaling cascade that it's called EGF-ERK signaling cascade. EGF is a molecule that attaches on the surface of the cell, and it is attached to certain receptors, and then when this thing happens, it generates signaling events of proteins, reacting with each other, and the final product is this protein here. It's called the ERK protein, and it has a PPP here because it's a protein that is doubly phosphorylated. In other words, there is a chemical part that has been added to the actual ERK protein in terms of two phosphate groups. And when this thing is done, this protein becomes activated and can go inside the nucleus, attach itself on the DNA, and generate activities inside the nucleus of the cells. And the results of these activities can lead to apoptosis, death of cells, can lead to cell proliferation, and some other things. Okay? Now, So, from a biological perspective, we may say that we are interested in this, in the concentration of this molecule. We don't care what else happens here, okay? As long as we focus on this molecule, because this is what eventually will affect the behavior of the cells. Of course, all of these things are simplified because our cells are very complex, and this is a small part of a very complex network. But anyway, we can see it this way. As a matter of fact, there is biological work has been done, experimental work, that shows that, okay, I will come back to this. Oh no, maybe not. Let's go back to this. So now, we know, so all of these things here are the molecules in our system, the molecular species. And this system has 23 different molecular species. They're all proteins and various versions of these proteins in terms of phosphorylation, single phosphorylation, double phosphorylation. This means that the doubly phosphorylated MEC has bound on this molecule, which is called a phosphatase, okay, and so on. So we have 23 molecular species. We know how to interact with each other because we have these arrows. For example, it says that this molecule, after a reaction occurs, which is a monomolecular reaction, and produces as a result this and this. Or this means that this reaction is reversible. This molecule can be as a reactant that will produce these two molecules, or these two molecules will be reactants that they will produce this molecule. And of course, there are the rate constants associated with all these reactions. And we have 32 reactions for this particular example. Okay?
So you're observing that what you call the ELK?
Yeah.
Those are your observations, and you're trying to read the system.
To read the system in what sense?
In other words, identify the network there.
No. I'm not talking in this talk about network identification.
Okay.
So I know I have constructed this network.
Okay.
I have it.
Can you map it back to the equation?
Yeah, I will. So I have constructed this network. I know how many molecular species I have. I know how they interact with each other. Okay? And also I have done experiments where I know the values of these parameters.
Okay.
So if I know everything, right, I can describe this thing by means of differential equations, which will be which will be of this form.
Right.
Okay? And then I can, sorry, I forgot something. This is also a matrix called the stoichiometry matrix that basically characterizes for a particular reaction, which is a product and which are the reactants and which are the products. So this is something also that, it's the stoichiometry of the network. So knowing the network, you know everything about here. You write this on a MATLAB program, and then you simulate it, and then you're going to come up with results that they will look like this. So for each species, you will plot the concentration of the species as a function of time.
For example, the representations that you showed for the equations, that's kind of akin to like mass action kinetics.
Yes, this is what they are.
Now, there are a lot more complicated kinetics like Michaelis-Menten or irreversible Michaelis-Menten that have high nonlinearities associated with them. What is the impact of understanding these characteristics when the representation is a little more complex?
First of all, my talk is on sensitivity analysis. So whatever I'm going to say here can be applied to any network model that you come up with. I just showed to you the one that we used for this particular network, just as an example. So as far as complexity in terms of equations, it doesn't affect whatever I'm going to tell you about sensitivity analysis. In terms of the inverse problem of identifying the network for measurements, there we can talk for hours. I mean, this is a very difficult problem that has many components, and all these things are very relevant. But here I'm talking about sensitivity analysis.
Okay.
So, you know everything about the network. That's the bottom line. Okay? So you can write the differential equations, you can solve them, and you can computationally evaluate the dynamic evolution of the concentrations of each one of the species as a function of time. That's fine. But now let's go to sensitivity analysis. The sensitivity analysis assumes that you have some output of interest. And again, you can pick up any of these concentrations or all of them, but all of them you may not have to do that, and complicates matters quite dramatically, because you are going to have to deal with 23 concentrations, and you have to track down how these concentrations change as a function of the parameters. But for this particular system, and for a particular biological problem, you are interested in this response for biological result I explained to you, which is basically this one here. Okay, so let's say that this is our output that we're interested in. Now, the problem of sensitivity analysis is this. If I change these parameters, the kappa parameters, the rate constants, how these changes are going to affect the output. So if I find rate constants that small changes produce large changes in the output, and I find some other parameters that large changes, they don't produce any meaningful changes in the output, then the second set of parameters will give me the idea that the reactions associated with these parameters are not very important. So the system is robust to those reactions. But in the first group, the reactions associated with these parameters are very crucial. This is what sensitivity analysis tries to do in this framework. Okay? But in order to proceed, we need to come up with some characteristics of these responses, so we can quantify the changes that occur. Okay? So there are three things that you can come up with that are explained by means of this figure. And it's the timing, duration, and strength of this response. Now, there are clear mathematical equations that you can use to evaluate this timing, duration, and strength with no problem. So you can, as long as you have the response, you can evaluate these quantities on the computer. The bottom line is that the timing tells you roughly when the maximum of the response occurs in time. The duration tells you how, the word says it all, how long this response occurs, and the strength tells you the amplitude. Mathematically, what you do is that you build up a parallelogram that is centered at the duration, at the timing that is calculated by means of this equation, and that this area is the same as the area of the response. So now you have three numerical quantities, the timing, duration, and strength, that they depend on the parameters of the system, on k, which given k, and of course knowing the stoichiometry and everything else, given k, you can calculate them. Okay? So sensitivity analysis will be based on numerically evaluating what is the effect of variations in the parameters k to these three responses. Now you're going to say, how do you pick up this? First of all, I didn't pick this thing up, somebody else did. But then it turned out that several people did experiments, biological experiments, where they showed that the timing, duration, and strength of ERK-PP directly affects biological function in terms of apoptosis, proliferation, and things like that. And that these are very, very important components as far as ERK-PP is concerned. So the bottom line is this, the moral of the story is this. Me, as an electrical engineer, as a computer scientist, or as a computational person, I will try to mathematically formulate things, come up with quantities that I can evaluate using a computer program, and there are many possibilities for that, but I have to constrain my possibilities based on biological evidence and experimental biological evidence. So for this particular problem, I have a very concrete biological evidence, because if I write a paper, I can reference 3-4 biological papers where they evaluate by means of experiments that these are three important quantities. So as long as I have set up the problem, now I'm ready to go with sensitivity analysis. So this is the idea. So what people do now? Of course, the easiest and the most straightforward thing to do is basically to, okay, so now you have a function called the response, which is either the timing or the duration or the strength in the previous example, which depends on k. For various reasons, actually for just one reason, I will show you here. You like to take the logarithm of the response, okay? But let's take the response. So sensitivity analysis can be done very easily by calculating the derivative of the response with respect to the parameters. Everybody can come up with this in milliseconds. Okay. Now, if you do that, though, it turns out that the kinetic parameters, the units of the kinetic parameters, are different in a biochemical reaction network, are different based on whether the reaction is monomolecular or bimolecular. So if you use the derivative with respect to K, then these derivatives will have different units, and that's not desirable. So to normalize them, you take the derivative of the logarithm of the response with respect to the logarithm of the parameters. And this is equivalent to this, as you realize this quantity is unitless. And this is called the local sensitivity index that you can calculate. It's called local because in order to calculate it, you have to do this derivative with respect to some given reference value for K. And because you are talking about derivatives now, for the derivatives to be accurate, the derivatives should be done by means of infinitesimally changing the values of K. You cannot change the case arbitrarily. However, in practice, how much you can change the case depends on how fast or how slow this thing changes a function of K. And taking the derivative, even if Rk changes fast, taking the logarithm of the Rk makes the variation slower. So it allows you to approximate these derivatives by taking appreciable differences between the parameters, if you like. But it depends on the application. Anyway, this is called the local sensitivity index. It's used extensively in the literature. And then by using this, we can quantify changes in the system response that are caused by infinitesimal changes in the kinetic parameters. And then we can run the reactions based on the value on the local sensitivity index values. If the local sensitivity index values are large, then the kinetic parameters associated with these indices are the most crucial ones. If the derivatives are small, then the kinetic parameter, the system is robust to the values of those kinetic parameters.
So experimentally, are these K observable or reachable?
Experimentally?
Yeah.
Yeah, you can change, but it's a very difficult problem. It's not very easy thing to do. Mm-hmm. But yeah, you can inject into solutions enzymes that they can increase the values of K. You can mask the activity of proteins. You can affect the rate by decreasing the concentration of the reactants. Because basically you're interested in the product of K times the concentration. So if you decrease or increase the concentration of reactants, it's like affecting the K. You can do things like that. Yeah. But so this is one thing. But there is another thing that you have the model now built by going through an inverse problem and so on. And now you have the model on your computer and you don't care about experimental stuff anymore. You just want to evaluate your model. Right. And come up with this. And when you identify the components, if your model is right and you have done it right, then you can go to the lab and focus on those components. Okay. There's some other stuff that are very new to us now, which change some of these things that I'm telling you. But I don't want to overwhelm you. This is the easiest way to present things right now. But let's say this will happen. I don't know if it will happen in our lifetime. But it will eventually happen. In pharmacology, people will develop a drug, okay, based on these models at some point, okay, by building up the mathematical models, by doing through sensitivity analysis, by identifying the crucial components, and then by going and developing a drug that is going to go and affect that crucial component, okay. And actually, companies have tried to do that. Vioxx was one unsuccessful and fatal attempt to do that. But they found out that there is a protein called COX-2 that if you reduce it, then if you change its concentration, then you're going to help patients with osteoarthritis. Okay? And this has been done, the information was obtained genetically by doing experiments and by building some models. However, it turns out that COX-2 also affects other things in the body, and some people have died because of heart attacks. So, the problem is complicated. But still, we're at the initial steps of that, and eventually, after several decades or, I don't know, centuries, we're going to reach this point, okay? So, this is the easiest thing to do. And if I do this on the model, I can make plots like this. This is the number of reactions. This is the sensitivity, actually the derivative of the timing, duration, and strength, the value of that index with respect to the kinetic parameter of that particular reaction. Okay? So, look what happens here. For most reactions, the sensitivity is negligible for all these three quantities, and we expect that because our cellular systems or networks are very robust. Again, for reasons that we all understand. But there are some reactions which produce very large sensitivity. For example, the sixth reaction produces a very large strength sensitivity. The amazing thing is that several months ago I went to Houston and I gave a talk, and there were some people from MD Anderson that they have been, actually a guy gave a talk before me and his results were experimental, and it just turned out that he was working on the same system, and turned out and he said, I discovered that this reaction was the most sensitive, and it was the reaction 6, as this model predicted computationally. Of course, the model is accurate because it has been published, the literature has been understood. So you expect this, but Okay, so this is what you can get. Now you're going to say, why don't you use this and let's go home now. Let's not do research, let's not continue with this talk. This is the easiest thing to do. However, it has several drawbacks. It's only a local assessment of sensitivity. You take derivatives, so the variations on the parameters are very limited, very locally around the reference values that you use. Depends also on the reference values that you use, and if you use different reference values, you may get different results, depending, of course, on how the response changes as a function of k. It's informative only in a small neighborhood around these values. The response functions are highly nonlinear, so you expect this behavior. So these are some drawbacks which you cannot evaluate very easily if you don't know much about your system. But one problem that you have is this, that if you want to calculate these derivatives rigorously without approximating them by means of finite differences or whatever, you can do that. Because you can go to the original differential equations, differentiate them on both sides with respect to k, and you are going to come up with another set of differential equations that will be in terms of the derivatives, of the sensitivity indices. So you can come up with differential equations that describe the evolution of the sensitivities. However, these equations are coupled together with the original differential equations, so now you have a larger system of differential equations to solve, and that's okay if the system is not crazy. But the problem is that this system of differential equations turns out to be stiff. Stiffness translates to the fact that you cannot get a result in a reasonable time. Theoretically speaking or mathematically speaking, stiffness is a very serious problem, because basically what you try to do is that you have a system of differential equations with some components that they vary very fast, these are the derivatives, and some components varying very slowly, which are maybe the concentrations, and you try to evaluate accurately both of these quantities in a reasonable time. So you're going to end up spending most of your time trying to estimate precisely the things that they change fast, and this may be overwhelming.
That's actually the question I had. Now, you talked about 32 reactions or something
Yeah,
like that?
for this example.
Yeah, in this system, is there a sequential pattern to it, or are all these simultaneous? What is the?
No, they're all simultaneous. You don't know how you don't know anything. There is no pattern here. in general. Okay, so you have to solve all these equations simultaneously.
So the coupling may be.
Yeah. Let me tell you what happened to me, because I did most of the simulations, so I have a first-hand experience of what happens. You know, the reference values that I used have been published in the literature, so I took them from the literature. I was not crazy enough to go to a lab and do experiments to estimate the parameters. I mean, a lot of people have worked for 10 plus years to get all these parameters. So the parameters were published. That's easy. You get them, you put them there, and fine. Now, I changed one parameter very slightly. So I ran the MATLAB program with the published parameters, and I got this figure. I didn't have a problem. It took me on this computer, I don't remember, like 10 seconds or something, okay? And then I took one parameter, I don't remember which one, some parameter, and I changed it very, very slightly. I increased it by 5% or 1%, I don't remember, very slightly, okay? And I couldn't get an answer from the differential equation solver. I was waiting, I thought, how long is it going to take? So I waited for half an hour. I just left it there working, and half an hour, no answer. Which of course proved the fact that the system is very stiff. But it proved something amazing to me, that the published parameters, for some reason, allows you to do sensitivity analysis, and if you change them a little bit, you cannot do it. So I don't know how these people come up with these parameters. I mean, it's a coincidence. I don't know. It may mean something. I have no idea, but that was very striking to me. Okay? So this is what happens here. So stiffness is a big problem. What did I do here? Okay. So now all of these things are very known, although this approach is used extensively in biological papers because it's the easiest to understand and the easiest to implement. However, we have to deal with all these problems. The other problem that bothers me, and I think it should bother any serious person in biophysics, is that this approach to sensitivity analysis, it's arbitrary, it's just a mathematical construct. It's not derived from basic biophysical principles. Now you're going to say, why do you care about this? Well, I made a thought in my mind before I went to Houston, but I was not daring to communicate the thought to anybody because I'm not an evolutionary biologist. I'm not even a biologist, okay? So I don't want to open my mouth and say things that logically may make sense, but biologically may be garbage. So my thought was this, that through evolution, perhaps our cells employed sensitivity analysis to build up their genetics. Okay?
Sir,
Yeah.
I have a question with point 3. It mentioned that you have to consider a very small neighborhood for nonlinear response function. Suppose we have a very large system and we are considering the small neighborhoods like 14 or 15 neighborhoods maybe. So is it possible to relate all the neighborhood to get a combined result for your system, or you can interpret the overall system with the results of all neighborhoods?
That's a good question, and you can say that, fine, basically you are saying that I can change my reference points and do this. Yeah, but I just told you also that by changing my reference point a little bit I cannot solve the equations. So the system is very stiff. Of course, I'm going to show you an approximation, a finite difference approximation, which is going to resolve this problem, and then it's possible to do what you're saying. But then whatever you do there is going to be arbitrary. It's not going to be mathematically rigorous. You're going to pick up different points, reference points, you're going to approximate the derivatives, you're going to get all these results, and now you have to combine them together. And how are you going to combine them together? You may say, I will take the average. All of these things are ad hoc. But there is a hope that you can come up with a sensitivity analysis which is biophysically meaningful, that it's rigorous mathematically, and it leads to what you're saying. And this is what I'm going to show to you if I have time. Okay? So the biophysical thing is very important in my opinion, because that was my thought. Perhaps evolutionary, the cell has been using sensitivity analysis to adapt to design its network, its networks. In a very simplified way, look at this. It started millions of years ago with a particular network, and then it tried to vary a few things and see what's going to happen. Of course, one thing that can happen is the cell dies. It dies, it's gone. But if it doesn't die with this variation, then it will adopt the variation, and if it gets some benefit, it will adopt the variation, it will survive, and through survival of the fittest, it will evolve. That was my idea. It's simplified. I mean, this did not happen like this. That was my idea. So I go to Houston, and then I get a guy, a professor from University of Houston, was moving me around with his car, and he told me what he's doing. He was an evolutionary biologist. So I told him, you know, he told me that he loved my talk. So I didn't know why. So I asked him, and I told him about my thought. And he said, of course, this is what we're doing. This is what we're working on. This is the basic principle that we're working on. Okay? So this simple idea may have impact on evolutionary biology. So it has to be, so in my opinion, sensitivity analysis has to be formulated not by an ad hoc derivative, but by something that has some more meaning. It's more meaningful in biology and also comes from basic biophysical principles. Okay? Plus there is another problem which I'm not going to tell you here extensively. You know what happens, and actually you may be interested in that too. The rate parameters, the rate constants are not independent with each other. Okay? And it took me actually, I was saying to Professor Krim here, that it took me the entire summer to learn thermodynamics. I didn't know thermodynamics. I used to know when I was a student, but I had a very bad experience, so I never paid attention to it because I couldn't understand it. Okay, he told me also that he had the same experience, so maybe it's an engineering experience. Engineers cannot understand thermodynamics. I don't know. So I tried desperately to learn thermodynamics and develop an approach, a mathematically rigorous approach that can explain the thermodynamics of the systems, which after 4 or 5 months with 2 students and working and arguing and so on, finally we managed it. And you come up with equations that they relate these parameters. So these relationships are there. So you cannot take arbitrary derivatives and you say, you know, all of these things, they have biophysical meaning and they have serious meanings here, based on the thermodynamics, on physics, on biochemistry and so on. So this is what we try to do with sensitivity analysis, to couple all these things together and come up with a technique that makes sense. So for all these reasons, this may not be appropriate. You can solve this problem of stiffness by basically approximating these derivatives by means of finite differences, and you can come up with formulas like this, where now you don't derive differential equations for the derivatives, but you keep all the parameters constant. You calculate the response for parameter Km. You calculate the response by perturbing the parameter Km by some amount. You find the difference of the responses, and you come up with this index. And we call this the finite difference local sensitivity index. And for small perturbations, it can provide you very good results in terms of the derivatives without having to solve the differential equations, so you don't have a stiffness problem. Instead of solving the original differential equations together with the differential equations, which is a stiff problem, you solve the original equations twice, and this is doable. Fine, but they have this approach has all the problems that I mentioned before. Plus, if you want to be precise, if it makes sense, because in biology a lot of things that they make sense in computer science and engineering, they don't make too much sense. So I may ask for these estimators to be very, very precise, these estimates to be very precise, but this may not be relevant to biology. But in any case, it's very difficult to control the accuracy of these estimators for various reasons that have to do with numerical analysis. You can come up with a step that will give you good results, good accuracy for certain derivatives and mess up all the other derivatives. So you have a problem there. But anyway, this suffers with all the previous problems that I mentioned, but this is another approach. The thing is that to accommodate stochastic parameter fluctuations, how are we going to do that? And there are many reasons why you want to accommodate stochastic parameter fluctuations. You can say, I don't know the values of the parameters exactly, so I have some uncertainty about the parameters, so I will model this uncertainty by means of a random variable. You can come up with all kind of arguments, but for me the most important argument is this, that going back to this evolutionary problem, I think it turns out that variations in the system occur randomly. And I will give you an example, okay? As time passes, there are changes in our DNA. We want it or not. Okay? That's why we get diseases. That's why when you get older, you have more possibility to get cancer because of these changes in the DNA that occur as a function of time, because of environmental conditions, because of many things, including also because of natural things, because our cells divide every little while, and every time that the cells divide, our DNA divides. And this division process that produces two DNA from one DNA, this birth process of this multiplication of our DNA is prone to errors. And of course there are biological mechanisms that they try to dramatically reduce these errors, but the errors are not zero. Plus these processes may be affected by environmental conditions and they may not do the job right. The bottom line is that for various natural and unnatural reasons, our DNA changes even during our lifetime. So you can imagine how they change as a function of centuries and thousands of years and millions of years, okay? So it changes. The most common changes are very small, isolated changes in one building block of our DNA, which is called a nucleotide. This can change. This change may be fatal, may lead to a very serious disease, just one change. But let's not go so far. There are most of the changes, which are very minor, very small, they tend to produce the same type of a protein, but with different kinetic properties. So these single nucleotide changes may mathematically be mapped to variations in the rate constant of the reaction that this protein participates to. Okay? But these changes, we cannot measure them. I don't have any microscope, and also I don't have enough time in my life to go back a million years with a microscope and start recording all the changes that they change. And I will tell you that, you know, through this 1 million years, my DNA has changed here. This is impossible, right? So there is a huge uncertainty, and we can model this uncertainty by saying that this is stochastic variations. So that's why we want that. So people have tried to come up with stochastic techniques, and for other reasons too. Here is what we're going to do. So we're going to assume that the rate constants change randomly. We have to formulate a mathematical statistical model for these parameters. And then this stochastic variation of the parameters will produce stochastic variation in the output of the system, the R of k. So k now is a random variable. R of k is a random variable. So we were going to evaluate the amount of change at the output by calculating variances. Okay? So this is what we're going to do. So, and then we're going to call a reaction to be influential if random fluctuations in the value of this kinetic parameter results in noticeable fluctuations in the system response. And otherwise the reaction will be assumed to be non-influential. This is how we're going to do that. So people have tried to do that by making various assumptions. One big assumption is that the kinetic parameters are statistically independent, but they are not, as I told you. So to build up models for the statistical variation of the parameters, you have to be very careful. And my recent experience says that you have to understand thermodynamics and how these parameters are related to each other to build up the correct model. And we're in the process of doing that. I will show you something here. And people have tried to do this. So one way to do it is to take the logarithm of the response, which is a function of k, and approximate it by a Taylor series expansion about a reference kinetic point, a logarithm of k bar. And if you assume a sufficiently smooth log response around this logarithm of k bar, you can show this formula, that the variance of the logarithm response is approximately equal to the sum of these m variances, v1 plus v2 plus vm, where vm is given by means of this quantity. Is the variance of the logarithm of the kinetic parameter associated with the reaction m times this derivative squared. Okay? This is an approximate formula based on several assumptions, but you can derive it rigorously. It's no problem.
What's the physical?
No physical. Mathematical. Taylor series expansion.
No. I'm just trying to understand actually the fact that you're basically trying to kind of eliminate large variation in R of k.
Yeah.
Is
this
there
is one thing.
significance?
You try to remove large variations from RK, and you try to basically, when you take derivatives, to be very local, and combine them all together, you come up with a first-order Taylor-Series approximation. You can show this formula. This is all that you do. This is what you get. So if I have this, then the output variance is the sum of these partial variances due to variations of individual parameters. And then you can come up with a sensitivity index, which will be very simple. Will be basically the VM divided by the variance of the output. The variance of the output is the sum of the Vn, of the little v's. So this is what it is. And this gives you that. And this is called a derivative-based stochastic sensitivity index. Stochastic sensitivity index. Okay, fine. It's again a mathematical construct. And look what happens here now. First of all, you still have this. This is the local sensitivity index. So you have to calculate this, no matter what here. So you go back to the previous problems. On top of that, you have also to calculate these variances. Now, the variance of the parameter, you don't have to calculate it. This is a parameter that you specify for your sensitivity analysis, because you have to come up with a model for the logarithm of Km. And the variance is a parameter. The smaller the variance, the more local the sensitivity analysis is. The larger the variance is the more sensitivity you introduce to the system, the more variation you introduce to the system. So this is a user parameter, fine. But then every time you pick up this, you have to calculate this, the variance of the output. And this can be done by using Monte Carlo simulations. But now the complexity of the problem is dramatic, because you have to calculate the derivatives as before, plus you have to do Monte Carlo. And you will do it only if you get a benefit. But the only benefit that I see here is that you introduce something that can deal with stochastic variations of the parameters, being stuck with all the disadvantages of the previous index. This is what it is. But we're on the right track, though. So anyway, I don't want to go through this. Again, you have stiffness problems, all the problems that we talked about before, plus the fact that you have to do Monte Carlo estimation of the log response variance. So now I'm going to try to address all the things that are highlighted by yellow here. Okay? With something that will be more appropriate for this problem in some sense. So there is a result in the literature. And actually, I don't know, Hamid, you know about Mark random fields. It comes from there too. This is related to the Hamilton-Clifford theorem. But anyway, it always shows up. So there is a mathematical result that says the following. If you assume that all the kinetic parameters are statistically independent, but again, I told you they are not, but that's not a problem, because I can use thermodynamics and I can use some biophysical principles, and I can cast the problem in terms of parameters that they are independent. So my sensitivity analysis will be with respect not to the kappas, but to the other parameters. So this is we're in the process of doing that. So I'm not going to show to you this. Let's assume that the parameters are independent. Then you can show that the variance of the output is given by means of this expression, where the little VMs here is given by means of this formula. And I will explain to you. These VMs are given by means of this formula. And you can derive formulas for the rest. But now, what these things mean? First of all, let's see what this thing is here. Okay? This term here is the expected value of the response when you fix the kinetic parameter of the mth reaction. So you fix the kinetic parameter of the mth reaction, you vary the rest, and you take the expected value of the response with the rest with this variation. Okay? So now if you do that, whatever is left, it depends on the kinetic parameter of the mth reaction, which also varies randomly. So you take the variance of the result. And similar expressions here. Okay, so this is a formula that this is exact, and the reason why you need statistical independence is that although this is always positive because it's the variance of something, this may not be positive, and you need all of these terms to be positive to do sensitivity analysis. So the only way for these terms, all of these terms to be positive is for the parameters to be statistically dependent. That's why you need that. Otherwise, this formula can be derived for any set of kinetic parameters, for any, okay? But the positivity is not going to be there. So anyway, so we have this formula. So basically what we do with this formula is that we take the variation of the output and we decompose it into partial variations, the V's that you see here. So if you understand this material, then you can say that the contribution of the mth reaction to the log response variance is given by means of this term out of all this stuff, where the first term is the contribution of the mth reaction by itself. These terms are the contribution of the mth reaction with cooperation with each one of the other reactions, the pairwise cooperation, and so on and so forth. So this is the formula, okay? So by doing this, then we can come up with some very interesting indices. The first one is called TESI. It's the total effect sensitivity index. You take the contribution of the mth reaction to the output and you divide by the entire variance, which makes sense. And by using some very simple probability theory results in terms of conditional variances and conditional expectations, and you can show that it's written by means of this, where this symbol now here with a parenthesis means that you keep all the parameters of all reactions except the mth constant. Anyway, don't focus on these equations because we don't have time now to understand them. This is technical stuff. Anyway, you can write it like this, and these formulas are used for Monte Carlo estimation of these parameters. You cannot use these formulas for Monte Carlo. You have to write them like this. Fine. So you have this, and then it turns out that there is a very interesting result that that you can show that the reaction M is non-influential if and only if tau of M equals to 0. A result that was not true before mathematically. Here you have it in an if and only if condition. So if I calculate these indices for my network and I identify those taus that their values are small, by the way, the values of the tau is between 0 and 1. This is another nice thing, where in terms of the derivatives you don't know how big or how small they will become. Okay, so if you set up a threshold and everything below that threshold will correspond to non-influential reactions based on this. Okay? The next index is to take the singular contribution of the nth reaction and divide it by the total. Again, it can be written by a formula like this that I can use for Monte Carlo simulations. And this is called the single effect sensitivity index. And I can use this to quantify sensitivity by the singular fractional contribution of the reaction to the response variance. And then, what happened? And I can rank influential reactions using the Cesi values. The larger the Cesi value for a reaction is, the more influential the reaction will be. In addition, I can develop also this index that is called the joint effect sensitivity index, which is basically the tau minus sigma. So as long as I have the tau and the sigma, I can calculate the epsilon. And basically the epsilon quantifies the sensitivity by the joint fractional contribution of a reaction to the response variance. Okay? So a reaction can contribute to the output variance singularly, only by itself, or jointly with other reactions. This quantifies the joint, the Cesi, the sigma, the singular, and there are some quantifies the total contribution. This is what it is. So having these quantities, I can play a nice game here that is described by means of this table. I can pick up a threshold theta, which is a small number, close to 0, it's user defined, and then if the sigma value of the mth reaction is negligible and the epsilon is negligible, then that means that the reaction does not appreciably influence the system response. So I can classify it as non-influential reaction. If the sigma is above the threshold but the epsilon is negligible, That means that the reaction influences the system response, but only singularly. So I call it an S reaction, influential singularly. If the sigma is less than the threshold and the epsilon is above the threshold, then the reaction influences the system response jointly. So I call it a J reaction. And in the remaining, it influences both singularly and jointly. So by using this approach, not only I have and I can build a biophysically based way to evaluate sensitivity using a statistical model and evaluated how the statistical fluctuations affect output variance, but on top of that, I can get information that the reaction is influential or non-influential, plus if it is influential, I will know if it influences singularly, if the reaction influences the output only by itself or in cooperation with some other reaction. Okay? So this is the idea. Of course, to do there are some drawbacks. We have to calculate all these quantities, which they have to be done by using Monte Carlo simulation. And this uses 2L times n plus 1 system evaluations, which is the number of Monte Carlo iterations is the L, and the M is the number of reactions. You remember the number of reactions was 32. L may be about 5,000, 6,000. On this computer, for this system, it takes about 36 hours of CPU time. Good luck. Although, to tell you the truth, if you have a model that is precise for a biological system and you know that, then you can wait even for months to get a result like this because it will be extremely useful. But you know, still it is too much of CPU time. So, and of course the CPU time also increases for larger parameter fluctuations. So there are some research challenges here that I mentioned. I don't have time now to go through this. Let me show to you this is related to something that is interesting, but okay. So now the question is that can we make the technique faster? So this is one research challenge. The other research challenge is this: how can we identify parameters that are statistically independent? The kappas are not statistically independent, so we have to identify parameters that are statistically independent. And of course, the challenge with this is to do that using basic biochemical and biophysical principles, using biochemistry, not using some abstract thing that we come as mathematicians or engineers or as computer scientists. And some other things that I don't have time to tell you now. But let's go back to making the technique faster, because this is also a very challenging problem. So the idea here is that can we approximate the response function with some other function that we can use it in these formulas to evaluate the sensitivity in the indices fast? So we're working on this. It may require spline approximations, all kind of things, but it has to be done right. We have a result which is very simple. I can approximate this with a function which has a settled form described by these equations. Okay, let me not go through this now. And the resulting approximation can approximate very well the sigma parameters, but not the tau parameters. Okay? And it uses Lagrange interpolation together with Gauss-Hermite integration, and eventually leads to a very efficient numerical technique for approximating the sigma values. And this is the result. This shows the actual result obtained by Monte Carlo simulation of the SESI, and this is the approximation. Okay? Pretty good. But this is very impressive. The first one requires 330,000 system evaluations or 36 hours of CPU time. The second one requires 160 system evaluations or 50 seconds of CPU time. But it's only for the SESIs, not the TESIs. So we've been working for many months now to come up with approximations, and we have approximations. But, you know, there's no time to talk about this. They are very complex, unfortunately, for explaining them. Okay, and there is a lot of effort that has been done in mechanical engineering towards this direction. A lot of effort, because they use it a lot. They use a lot of sensitivity analysis there, but not variance-based sensitivity analysis. They use other, but they still have to approximate things. Okay, so these are some other results for different levels of fluctuation. So this is 10% fluctuations of the parameters. This is 20% fluctuation of the parameters, and the results are very consistent. But if you go above 30% fluctuations, then the approximation fails, because the original function is nonlinear. So the variation over larger fluctuations will be more difficult to approximate it. Okay? So we're working on this kind of challenges. So the conclusions are that it seems like variance-based sensitivity analysis is very effective, both from a theoretical perspective, from biological perspective, and for a perspective of explaining how things should be studied in terms of sensitivity analysis, but it's computationally very intensive because it requires extensive Monte Carlo simulations. Results in derivative-free sensitivity analysis, and has all these benefits associated with something that does not use derivatives, can be applied to a wide range of parameter values. And in this talk I have some arguments to show to you that you can do also global analysis based on this approach, which is under development now, and may lead also to rigorous statistical and information theoretic approaches, because by having a statistical approach and by calculating variance expectations, you can build up hypothesis testing techniques to make sensitivity analysis more statistically rigorous, instead of doing arbitrary things or ad hoc things. And one of the major challenges is to come up with computational efficient estimation approaches for these indices. Okay, and that concludes my talk.
Okay, we have time for quick questions. You can ask him your questions offline. Just a quick, one or two quick questions. Running a bit late here.
Okay, that's good.
All right, let's thank our speaker again. So I'm going to leave you in care of Kranos for the next